Hello, my dear students. Welcome to my channel, Patel Science Academy. Dear students, you are well familiar with me. This is your Shahzad Yunus Patel, sir. And dear students, in this lecture, you will be able to know about the topic DNA packaging in the eukaryotic cell from the chapter Molecular Basis of Inheritance. Dear students, you are well familiar with this chapter and with this academy. Dear students, let's start the topic without wasting the time. DNA packaging in the eukaryotes. Dear students, what is eukaryotes? Eukaryotes are those of living organisms in which the eukaryotic cell is there. Their body is made up of eukaryotic cell. And dear students, what is eukaryotic cell? The eukaryotic cell are those cell which contain the well organized nucleus. You can see here the well organized nucleus is found in the body of the eukaryotes, because their body is made up of eukaryotic cell, and it contains the nuclear membrane and nucleolus and the thread like material in the form of the chromosome. So dear student, this is the very first point of eukaryotes. And because we are going to know about the eukaryotic cell DNA packaging, so you have to be very familiar with this topic. Now the second point regarding the DNA, regarding the eukaryotes, it includes the chromosomes. DNA is associated with the histone protein and the non-histone protein. Dear students, these are two topics in which is related to the DNA. One is the histone protein and another one is the non-histone protein, and these histone and non-histone protein was reported by the R. Kornberg in 1974. So dear students, you can note down all these points one by one. Here we have seen 2 points regarding the eukaryotes. Point number one, it includes the eukaryotes, which shows the well organized nucleus and which contains the nuclear membrane, dear students, nucleolus, and the thread like material that forms the chromosomes. And one more thing regarding this topic, the DNA in this cell, the chromosomes, DNA is always associated with the histone protein and non-histone protein, which is reported by the R. Kornberg in 1974. Dear students, now we'll see the point number third. It includes the organization of the DNA is much more complex in the eukaryotes. As you are well familiar with this point, the organization of the DNA is much more complex in the eukaryotes than in the prokaryote cell. The next point, dear students, next point includes the topic, the point, depending upon the abundance of the amino acids residue with the charge side chain. Dear students, a protein acquires its charge. So it depends upon the abundance of the amino acids residues with the charge side chain. Dear students, next point in this topic we'll see is the regarding the histone protein. And these histone protein are the protein that are rich in the two amino acids residues. And what are the name of that two amino acids residues? Number one is lysine and number two is arginine. So you can see here lysine and arginine, these are the two amino acids residues which are richly present in this histone protein. And about this packaging of DNA, it is totally regarding the histone protein. And yes students, there is a big role of the histone protein in the DNA packaging in the eukaryotic cell. Dear students, both these amino acids residue, which one? The lysine and the arginine, we have seen in the point number 5. These are the basic amino acids and carry the positive charge with them. So dear students, what about this positive charge? This positive charge is present on the amino acids residues, which one is lysine and arginine, on the histone protein. And dear students, as you are well familiar with the DNA is always having the negative charge. So negative charge is on the DNA and positive charge is with this histone protein to lysine and arginine residues of this amino acid. So there will be a definitely combination in between this histone protein and the uh, DNA molecules. So there is a big binding between the DNA protein and the DNA and the histone protein. So histones are the sets of positively charged basic proteins. Means there is positive charge arginine and lysine residues of amino acids in the histone protein. So it clears that uh, histone protein shows the positive charge and DNA shows the negative charge. So this positive and negative charge combination makes a compound there, and that compound is nothing but the binding of the DNA molecules with this protein, histone protein. So it is called a packaging because it get packed with this histone protein, and these histone organize themselves to make the unit of the 8 molecules. And this is the most important point of this slide, dear students. Point number 8. What is that point number 8, dear students? This is the histone, these histone protein which is containing this lysine and this arginine. This histone protein, dear students, they organize themselves and make a unit of the 8 molecules. How many molecules? 8 molecules unit is made by this histone themselves, and these 8 molecules, because 8 molecules are there, this histone is known as the octamer, because octa means 8. So all these histone protein together make octamer. And what is that octamer? We'll see its structure now. Dear students, as this slide is only for the packaging of the eukaryotes, so you can note down all the points one by one. Here I have explained you about the 8 points you are watching here right now. So in the first point we have seen eukaryotes are the well organized cell, and it contained well organized nucleus and nuclear membrane, nucleolus, thread like material, etc., and chromosomes. And it was associated it is associated with the histone and non-histone protein. And dear student, it consists of two molecules of amino acids residues, and that two molecules are lysine and arginine. This is most important point in this slides. Means histones is rich in the lysine and arginine residues, and because of this lysine and arginine amino acid residues, it shows the positive charge, and DNA is having negative charge. So positive charge and negative charge, these two molecules comes together and packed with each other, and this histone organize themselves to make the unit of the 8 molecules. And how many molecules are there? There are 8 molecules, and this is called histone octamer. And the structure of the histone octamer, now we'll see, dear students, the structure of the histone octamer together with the DNA. It means there is a formation of one structure. That structure is called nucleosomes, which is in front of you. What is nucleosome? Nucleosome means the histone octamer plus DNA molecules. So if you are confused regarding the nucleosomes this term, so just look at here, the histone octamer and the DNA molecule together, it is called nucleosome. So this is new term I'm introducing here in the packaging of the DNA in the eukaryotic cell, dear students. So what are the topic uh, we are watching here in this structure? Nucleosomes. What is nucleosomes? Nucleosomes, the combination of this octamer, the structure which is made up of these 8 molecules of histone protein. And you are watching here these 8 balls, and 8 balls indicates the octamer of the histone. And just let me zoom it and let me explain you about this octamer in this structure. Dear students, you will be able to see this octamer here. This yellow color 8 balls are the histone molecules uh, and actually these histone molecules, in this histone molecules you will be watching here H2A, H2B, H3 and H4. So each of these molecules are in 2 in number. So 2 molecules of H2A, 2 molecules of H2B, 2 molecules of H3 and 2 molecules of H4. Likewise, all these 4 molecules into 2, it shows the 8 molecule structure you are watching here right now, and this 8 molecule structure is called octamer of the histone. So this is 1 2 3 4 5 6 7 and 8. So likewise these are 8 molecules of the histone octamers. And what are there in the 8 molecules? 2 molecules of H2A, 2 molecules of H2B, 2 molecules of H3, 2 molecules of H4 histone protein. But what is nucleosomes? So dear students, as I told you, nucleosomes is the histone octamer plus DNA molecules. So uh, you are watching here, this yellow color balls are the histone octamers, and what is this orange color structure? This orange color structure which is wrapped around it, around this histone octamer, this is DNA, you can see here. So DNA plus this histone octamer together, it is called nucleosomes, and this is the H1 histone which continues this histone octamer, which is means the nucleosomes with the another nucleosome. So this is the linker DNA, it links this nucleosomes with the another nucleosomes. So dear students, I hope you are getting it and you have understood about this topic, the nucleosomes. What is nucleosomes? The nucleosomes means the combination or mixture of the histone octamer plus DNA molecules. What is histone octamer? The 8 molecular structure of the histone protein. How many histone proteins are there? The 2H2A histone protein, 2H2B histone protein, 2 molecules of H3 histone protein and 2 molecules of H4 histone proteins. All these together in the form of these 8 balls is known as histone octamer, and you are watching here the DNA molecules in the orange color structure are wrapped around it. So these two structure, histone octamer plus DNA, is called nucleosomes. So we have seen here about the nucleosomes. Now we'll see the detailed account of this nucleosomes with the octamer structure. The negatively, dear students, this negatively charged helical DNA, this negatively charged helical DNA is wrapped around the positively charged histone octamer. As I told you, histone is a positively charged structure and DNA is a negatively charged structure. So this negatively charged helical DNA is wrapped around the positively charged histone octamer, forming a structure. What is that? What is the name of that structure? As I told you, this is the combination of histone protein, which is positively charged, and the DNA molecules, which is negatively charged, and this is called nucleosomes. I hope you are getting it, dear students. The second point from this histone protein and the DNA molecules together known as nucleosomes. The nucleosomes core is made up of 2 molecules of each of 4 types of histone protein. As I told you, 2 molecules of H2A, 2 molecules of H2B, 2 molecules of H3 and 2 molecules of H4. It clears that in the octamer there are 4 types of histone protein: H2A, H2B, H3 and H4, and all of these 4 types of histone protein, it shows there are 2 copies in that structure. That's why these become 8 in number, and likewise this is the octamer of the histone protein. Dear students, this H1, what was H1 there? As you can see here in this structure, this green color part, what is this green color part? It is given a name H1 histone. So we'll see about this H1 histone structure here. What is this H1 histone in this number 3 point? This H1 protein is the protein which binds the DNA thread where it enters or arrives and leaves the nucleosomes. Means the entry of the DNA thread in the nucleosomes is possible because of this H1 proteins. So H1 protein, dear students, this H1 protein, it binds the DNA thread where it enters and leaves. Means this DNA molecules can enter into the nucleosomes by this H1 protein, and it leaves also this nucleosomes for entering into the next nucleosomes. So this is about this structure of nucleosomes. One more point, one molecules approximately contain 200 base pairs of the long DNA helix which bound around it. So how many base pairs are there in the DNA molecules? One in the one nucleosomes. This is very important point, dear students. There are 200 base pairs in this structure in the nucleosome. In the one nucleosomes there are 200 base pairs, and about dear students, you can also see the last point is again very important. About 146 base pairs. How many base pairs? 146 base pairs long segments of the DNA remains present in each nucleosomes. So here two important points you can see. Point number 4 as a most important point. In the one nucleosomes about 200 base pairs are there, and about 146 base pairs long segments DNA remains present in each nucleosomes. What is H1? H1 protein binds the DNA thread which enters and leaves the nucleosomes. So this is about this slide, dear students. Now we'll see the next point from this nucleosomes as a point number 6. Dear student, these nucleosomes are the repeating units of the chromatin. Now we'll introduce the next point again, the next term. In the previous lecture we have introduced in the DNA packaging in the eukaryote cells nucleosomes. Now we'll see one more term here. Nucleosomes are the repeating units of the chromatins. Means this chromatin is the new word, and when this nucleosomes repeats itself one by one, so it makes a structure known as chromatin. And dear students, which are thread like stained or colored bodies present in the nucleus, and this looks like a beads on the string when observed under the electron microscope. So what is the new term now you are watching here in this structure, in this slides? The chromatin. What is chromatin? The nucleosomes, which is the combination of the DNA molecules and the histone octamers. It repeats itself, and it is the repeating unit of this structure you are watching here, and it is called chromatin. And it look like the beads on the uh, electron under the electron microscope. So if you are confused regarding this beads of string, so you can see here this structure I am drawing here, dear students. You can see here the beads of strings. This is the beads of strings. For example, this is here DNA molecules, and these are nucleosomes, different nucleosomes. These are different nucleosomes, and the structure in this shape is called the beads on the strings. Because you can you can take the another example. The women's wear the necklace, and that necklace shows the diamond, and that different diamond arrangement in the necklace is like the beads on the string. You can consider it likewise. And originally I'll explain here this structure. You can see here this structure, dear students. This is that double helical structure of the DNA. This is the double helical, two helix, helix number 1, helix number 2, and this is called DNA. And dear students, this DNA, it makes nucleosomes, as I told you, and this is that nucleosomes, one of the examples of the nucleosomes you are watching here. And this you can consider this structure as a nucleosomes. Means this is the uh, what we say 8 molecules structure of the histone protein, that is called octamer plus DNA. So it is always known as nucleosomes. This nucleosomes together, this is one nucleosome, this is another one nucleosome, this is one more nucleosome. So this structure is likewise it assumes the shape of this structure, and it look like some beads on the string in which DNA wound around the nucleosome. So it look like that, and in this next point it is looking like that, which is called the chromatin fibers. So this is the beads of strings, the arrangement of the DNA on the nucleosomes, and this is the continuation of this nucleosomes after having wrapped on the octamer, and after entering into one nucleosomes it goes for the next nucleosomes, and from next nucleosomes it goes into the another nucleosomes, and likewise it goes for the next nucleosomes, and it assumes the structure which is known as the bits of strings. So this is about this bits of string, which is the chromatin, which is the actually the repeating unit of the nucleosomes. Yeah students, this is that chromatin showing the bits on the strings. You can see here, these are the nucleosomes. This dot 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 you can see here, this dot indicates the nucleosomes, and different dots are linked with this thread like structure. This thread is a DNA, and this dots you can consider as nucleosomes. It is actually nucleosomes, and this nucleosomes with this DNA is continuous, and its length you can see here 0.5 mm, and this is the structure known as the bits on string under the microscope. Dear students, now we'll see the next diagram. In the next diagram you can see the zoomed part of this bits on the string in the magnified form. So in this structure you can see the three nucleosomes are there. This is nucleosome number 1, this is nucleosome number 2, this is nucleosome number 3. And what is this red color thread like structure, which after assuming some distance it wrapped over this nucleosomes, and this is that linker DNA which continues this on one nucleosomes, and it allows it to enter into the next nucleosomes. And you can see here, dear students, these are the wound these DNA wound around the histone protein. So this is nucleosome number 1, this is nucleosome number 2, this is nucleosome number 3, and this structure it look like beads on the string, like the diamonds in the necklace of the women's. So this is the beads on the string, and I think you are not confused regarding the bits on string. First we have seen about the DNA double helical structure in the eukaryotic cell. Then we have seen about the histone protein. Then we have seen about the octamer, the 8 molecular structure of the histone protein. Then we have seen the nucleosomes, the octamer with the DNA wrapped structure. Then we have seen the different nucleosomes repeats itself and forms the chromatin, and it looks like this beads on the string. Now we'll go for the next structure, and next structure is the DNA helix. It consists of the 200 base pairs wraps around the histone octamers by one three fourth turns, and 6 such nucleosomes get coiled. How many such structures? 6 such structures of DNA molecules which consist of, or which contain 200 base pairs, which is wrapped around the histone molecule octamer. These 6 such nucleosomes get coiled. How many nucleosomes? 6 nucleosomes get coiled and then form a structure. That structure, dear student, is known as solenoid. So what is solenoid? Here one more time I am introducing one more new term, solenoid. So solenoid is the mixture of not mixture of, it's a combination of 6 nucleosomes, dear students. So these 6 nucleosomes together form a structure that is called solenoid, and that looks like the telephone wire. As I hope you have seen the telephone wire, and solenoid is the term we use for that 6 molecules of nucleosomes which look like a telephone wire. So here in this structure you can see the solenoid structure. At the top there is a bits on the string. Thereafter it bits on the string is 11 nm in length. Then it forms 6 nucleosomes together forms the solenoid, and solenoids together forms the fiber, and fiber together form the chromosomes. So in this structure, this is that structure with wraps. I'll explain in the next slides you properly. Here you can see the structure of the solenoids. Where is solenoids, dear students? You can see this telephone wire like structure. This is called solenoid. So if you go from the beginning, so you can see this is histone protein, octamer, DNA is wrapped over it, it gets supercoiled, and we call that structure nucleosomes. This is nucleosomes. This is H1 protein I have explained, which binds this DNA with this histone octamer. This is another nucleosomes. You can see here this nucleosomes together forms the chromatin structure. I have also explained about it. And this 6 nucleosomes together forms the structure that is called solenoid, and this solenoid is the structure of this nucleosomes, which looks like a telephone wires, dear students. So I hope you are getting about the packaging of the DNA in the eukaryotic cell. Now we'll go for the next structure, next topic, that is the chromatin fiber, which is made by the solenoid. So definitely solenoid will condensed and packed to form the solenoid. This chromatin structure, it is packed to form the solenoid structure of the 30 nanometer in diameter or 300 angstrom unit, and which gets super coiled and again coiled, which is called super coiled. So this solenoid structure super coiled and form the loop together, and this looped structure is called the chromatin fibers. So here you can also learn about this solenoid structure. Means the solenoid structure super coiled and form the chromatin fiber. So uh, this chromatin fiber is the structure made by the solenoid, which is having the diameter of 300 angstrom unit or 30 nanometer. Dear students, this is that chromatin fiber you can see here. So let me explain one more time. This is DNA helical structure which forms together the nucleosomes. Nucleosomes is the combination of this DNA molecules and the octamer, and together this is called nucleosomes. This nucleosomes it continues with the H1 proteins by the linker DNA, and it look like a bits on the string, and then it forms the 6 molecules of this nucleosomes together forms a solenoid structure, and solenoid structure super coiled together, and this is looking like that, and this is called chromatin fiber. And what after the chromatin fiber, dear students? This is the term chromatin fiber will definitely form the next structure, and that next structure is chromosomes, that we'll see in the slides. This chromatin fiber coils and condensed at the metaphase stage of the cell division and forms the chromosomes. So this is that chromosomes, as I have told you, at topmost structure. This is from the chromosome. So dear student, this is the play of this packaging of DNA. From the DNA helical structure, it is responsible for the formation of the chromosomes. So it is now clear that chromosomes, on the chromosomes there are DNA, because DNA, it is packed over the octamer histone protein, and histone protein is positively charged, DNA is negatively charged. Dear students, that's why there is a combination of the octamer and the negatively charged DNA molecules. And dear students, that structure is called, as you know, that structure is called nucleosomes. Different nucleosomes together, it looks like a beads on the strings, and that bits, 6 nucleosome structure together, it is called the solenoid, and solenoid together supercoiled and forms the chromatin fiber, and chromatin fiber then coiled and supercoiled and get condensed and form the chromosome structure. So DNA together with the histone protein forms nucleosomes, which looks like some beads on the string. New 6 nucleosomes together forms a structure called the solenoid, and this solenoid structure supercoiled together to form the chromatin fiber, and this chromatin fiber condensed and form the chromosomes, as you can see at top. So this is about that packaging of the DNA in the eukaryotic cell. Dear students, one more term is here, NHC, that stand for the non-histone chromosomal protein. What is this NHC? Dear students, the packaging of the chromatin at the higher levels needs an additional set of the protein, and that additional set of the protein which is needed for the packaging of this DNA on this chromosomes, it is called the NHC, that is the non-histone chromosomal protein. You can also remember this term, because at the higher levels this is also needed for the packaging, for maintaining this packaging of the DNA on the chromosomes. So this is about that NHC. Dear students, this is about this lecture. Thank you very much for watching this video. Dear students, one more time let me have the revision of this topic. First we have seen the chromatin, which shows the bits on the string, and DNA, which forms the which with the combination of this histone octamer makes the chromosome structure. It makes the structure of the nucleosomes together, it looks like the bits on the strings. This you can see the these bits on the string. One nucleosome is continuous with the linker DNA on the another nucleosomes, and it forms the structure, which structure is looking like the nucleosomes. Then this nucleosomes, it assumes the 60 angstrom unit this diameter, and chromatin fibers which stretched during the preparation of the for the electron microscopy, revealing the linker DNA between the nucleosome structure, you can see here. And this nucleosomes together, it forms this structure, and this structure, it shows the linker DNA as well as the nucleosome. This is the nucleosome score, dear students, and this linker DNA vary in the length from the 8 to 114 nucleotide pairs. And nucleosome scores, it consists of, as I told you previously, it shows the 144 nucleotides pairs of DNA wrapped 3-4 turns around the octamers of the histone, and this is the chain of the nucleosomes forming 10 to 11 nanometer thick fiber, which is called the chromatin fiber. Here students, thereafter, dear students, this structure is formed, and this structure is the solenoid structure or solenoid structure, forming a 30 nanometer thick fiber. And you can see here this 30 or 300 nanometer thick fiber structure, it is the metaphase fiber or the solenoid, and this is about 110 angstrom unit fiber. It is having this diameter, so this is called that solenoid, which is made by the 6 nucleosomes. So this is about this packaging of DNA, dear students. So packaging of DNA, we have seen about the packaging of the DNA. DNA is responsible for the formation, for wrapping on this structure. Now you can see this 8 structure. This 8 structure, it is showing the H2A, its 2 molecule, H2B, its 2 molecule, H3, its 2 molecule, and H4, its 2 molecule, like its 2 molecules. Likewise, there are 8 molecules of this histone protein, and this structure is called octamer, and DNA were wrapped around this octamer, and this structure is together called the nucleosomes. And this nucleosomes, it look like uh, uh, the beads on the string. Dear students, you can see this red color structure is the DNA, this green color structure is the H1 linker or H1 protein, and this is the histone protein octamer. You can see this octamer, 8 balls of H2A, H2B, H3, and H4. Each of 2 is showing here as a nucleosome. So you can see here the core of the 8 histone protein as a nucleosomes, and this nucleosome, 6 nucleosomes form this structure, as I have explained you in the previous slide. So I hope, dear students, you are well familiar with this structure now. And this is about this lecture. This is your Shada sir saying you goodbye. Stay home, stay safe, and don't forget to subscribe my channel, share, like, and comments for your queries. And dear students, please do comment for your queries, and uh, don't forget to subscribe my channel. Thank you very much for watching this video.